This here is a model of a semi-permeable plasma membrane found in the animal and plant cells. The role of the plasma membrane is to regulate the exchange of materials into and out of the cell. Phospholipid bilayer is the main structural component of the cell membrane, consisting of hydrophobic phospholipid tails and hydrophilic phospholipid heads, which allow the membrane to be fluid at room temperature. This fluidity moves at a rate of 2 micrometers a second. This is cholesterol, which is found within the bilayer and maintains fluidity at low temperatures. This is a type of integral protein which assists with the transportation of particles through the plasma membrane. This specific type of integral protein is called a carrier protein. This is a peripheral protein which exists on the perimeters of the cell membrane. This peripheral protein is also a glycoprotein, which are the basis of cell recognition and identification. It is a glycoprotein because it has an oligosaccharide carbon chain at the top, which regulates the cell recognition. This is a glycolipid which acts the very same way as the glycoprotein, except the oligosaccharide carbon chain is found at the top of a lipid. In this animation, we will go through two types of passive molecule transportation through the semi-permeable cell membrane. These are simple diffusion and facilitated diffusion. Simple diffusion is the passage of non-polar molecules from a high concentration area to a low concentration area in order to reach an equilibrium without the involvement of a membrane protein. Here we have oxygen molecules. In the extracellular fluid, there is a higher concentration of these molecules than there is within the cell. Therefore, these oxygen molecules will begin to move into the cell through the process of simple diffusion. Facilitated diffusion occurs down a concentration gradient, mirrored to simple diffusion. Although facilitated diffusion involves the passage of polar molecules, therefore they cannot simply pass through the phospholipid bilayer. They must utilize the assistance of carrier proteins in order to pass through the semi-permeable plasma membrane. These here are sucrose molecules. Sucrose molecules are polar and need a sucrose transporter protein in order to pass through the cell membrane. As you can see here also, there is a higher concentration of sucrose in the extracellular fluid than inside the cell. Therefore, sucrose will move into the cell in order to reach an equilibrium. Once the sucrose molecule is in the carrier protein, the protein configures its shape, opening a passage to the other side.